Hello, my name is Callie Price, and I will be giving this video for those of you that were unable to come to a makeup lab. We held makeup labs because Tuesday labs were canceled due to the weather. Please note, when looking at these videos that I'm going to post into your Blackboard account, I will also give instructions on what you need to do to still turn in your worksheet and get a grade. This first video will be an introduction of where I will go through the PowerPoint of which you also have in your Blackboard account. Please open and view this PowerPoint as well as view this video to make sure that you understand the material. If you do not understand anything I'm about to say through these videos, please contact your lab instructor before your practical test next week. I also want to announce that there will be a lab practical review on Friday, February 14th from 10 a.m. to 2 p.m. in Lab Room 220. This is come and go as you please. Lab instructors will be here to answer your questions that you might have. If you missed a lab, you can come look at the slides, the specimens, or the experiment that we did for that particular lab. If you are not able to come to a makeup lab this week, especially for Chapter 5, please try to come to the practical review and ask any questions that you might have. This week's lab covered Chapter 5, Prokaryotes. Prokaryotes are bacteria, which are microscopic organisms that are found in almost every environment on the planet. Prokaryotes have more diversity than any other organisms combined. There are anywhere from 500 to 1,000 species in the human body alone. The number of prokaryotes living in your body is greater than the number of human cells, or eukaryotic cells. I like this cartoon right here. It's a portrayal of a human compiled with many bacteria, and it says, "We are all just petri dishes with shoes." We classify prokaryotes into two different domains. We have domain Archaea, which are our extreme bacteria. They live in extreme environments, such as places with very hot temperatures or very cold temperatures. We also have the domain Bacteria. These are considered our regular, everyday bacteria. This is what we will be studying in today's lab. Now, most of these bacteria are actually not pathogenic. Pathogenic means disease-causing. Now, bacteria usually get a bad rap and you hear of them as being harmful, but they're actually only harmful if they get out of the environment that they're supposed to be in. For example, you always hear bad things on the news about E. coli, such as E. coli being ingested by humans, such as being on the spinach that they have eaten. This then makes the person sick. However, we also find E. coli in our intestine. E. coli is actually very beneficial inside of our intestine. It is only when E. coli gets out of our intestine onto our food and then re-ingested that it becomes harmful or disease-causing. So again, most of them are actually beneficial until they get out of the environment that they're supposed to be in. We have three tools that we can use to identify bacteria. The first tool is cellular morphology. This is where you would be looking at the bacteria through a microscope. The second tool are biochemical or physiological tests that we can use to identify bacteria. The third tool are colonial characteristics of bacteria. Let's start with our first tool, cellular morphology. Cellular morphology is where you would be looking at the bacteria through a microscope. In this week's lab, I gave a slide that had many bacteria on the slide that you needed to look at through the microscope. I told the students they needed to identify the shape and the grouping of the bacteria. For your practical, you will need to be able to look through a microscope that I have already focused in for you and either tell me the shape of the bacteria, the grouping of the bacteria, or both. For example, if you look at this picture up here, I have a rod-shaped bacteria. The rod-shaped bacteria is called bacillus. If we look at this picture here, I have round bacteria. If the bacteria has a round shape, we call this coccus. If the bacteria has a spiral shape, we call it spirillum. For the grouping, we have strepto or staphylo, and there's supposed to be a Y in there, as I've drawn in. Strepto means the bacteria are in a chain, one or more bacteria in a chain, such as this. We call it staphylo if the bacteria are clustered into groups, such as this picture right here. And again, I drew an example here. So if they are clustered together or piled together, we call it staphylo. If they are found one or more in a chain, such as this, we call it strepto. So once again, be able to identify the shape of the bacteria and the grouping of the bacteria for your practical exam. We did not take any measurements of the bacteria on this slide, so we will not use size. Again, please be able to look at the microscope and identify all these characteristics for cellular morphology. Our second tool that we can use to identify bacteria are biochemical or physiological. We will not use this tool in this lab. Just know that this is a tool that we can use to help identify bacteria. This tool would use different things such as the presence or absence of enzymes, or we could also use sugars to use as a testing material that ultimately we would end up testing the pH. But again, we will not use this test, just know that it is a tool we can use. Our third tool we can use to help identify bacteria are colonial characteristics. Colonial characteristics is when you would grow the bacteria on a petri dish like you did over this last week. The bacteria is grown on agar, as you can see this yellow substance in the dish. The agar is the medium of which the bacteria can grow on. For colonial characteristics, you would look at the size of the colony, the shape of the colony, such as does it have a rough edge, like this picture here, or does it have a smooth edge, such as this bacteria colony here. And then we can also use color. As you can see, they can be multiple colors. We can also look at appearance of the colony. So is it opaque, meaning we cannot see through it? Or is it translucent, meaning that it's clear or you can see through it? Now for colonial characteristics, to identify the type of bacteria, you would use what is called a dichotomous key. You have a dichotomous key in your lab manual in Chapter 5. You will need to know how to use this key for your practical exam. We grew 4 different species of bacteria for you to look at. We grew Bacillus subtilis, Serratia marcescens, Sarcina aurantica, and Enterobacter coli, or E. coli as you would know it. You will need to be able to use this key right here to be able to identify these species of bacteria that we grew for you. I will explain how to use this key in a separate video. The other part of this lab, we counted bacterial colonies that you grew on your surfaces over this past week. You chose 2 surfaces to swab and grow bacteria. You chose a surface A that you said was clean or would have less bacteria, and you swabbed a surface B that you called dirty or would have more bacteria. We let these bacterial colonies grow for a week, and then we took counts for both of these surfaces as a class. I will go over this worksheet that we did for these counts in a separate video. You will need to watch that video and complete your worksheet and turn it into your instructor next week to get a grade. That being said, I will continue this lab on the separate videos.